Hi, welcome to the BYU-Idaho Tutoring Center videos. Today we're going to be talking about the origins of genetic variation. So genetic variation is just the differences in members of a population or within a species. So you look around, you'll notice that everybody in our world basically looks different from one another. And this is genetic variation, these differences between members of the same population. And so how does this happen? Well, we're going to answer it using principles of meiosis. And keep in mind that this is applying to sexual life cycles because all of these are with meiosis, which only sexual organisms undergo. So the first one is independent assortment. Independent assortment describes the random alignment of homologous chromosomes on the metaphase plate during metaphase 1. So in this example right here, I'm going to show two different scenarios or ways in which the chromosomes could line up, and then thus the different combinations of gametes. So the red is going to represent the maternal DNA, or the stuff you got from your mom, and the blue is going to represent the paternal DNA. So each of these is a homologous pair. So the homologous pair could line up this way, or it could line up this way, flip-flopped. And so remember, so because of this, there's a 50% chance that the maternal is going to be on the right, or 50% chance that the maternal is going to be on the left. And remember that this is independent assortment, so each of these is going to sort differently. So just because the red's on the right in this side doesn't mean that it's going to be on the right in this homologous pair. And so because they're each independent, we're going to be able to create a lot of variation. So if they lined up this way, then we underwent meiosis 1, these would be our two daughter cells, and then when we underwent meiosis 2, these would be our gametes that were formed. And the same thing with this one. And so as a result, we would have kind of 4 different combinations that we could form just with 2 homologous pairs. And if you'll see those there, each of these 2 is the same. And so the way that we can calculate this, you might be asked on a test sometime, or a homework question, if we have 23 chromosomes, which would equate to 23 homologous pairs, how many different combinations could we form using independent assortment? So what you're going to do is you're going to take 2 to the nth power, and n is just the number of homologous chromosomes. So example, we had 2 homologous chromosomes, or pairs of homologous chromosomes, so 2 to the n is 4, and we see that we got 4 different combinations. So that's the first way that we create genetic variation within a population. Because now each of these gametes has different genes or chromosomes which will code for different things, which will result in genetic variation. The next one is crossing over. So crossing over happens in prophase 1, and it occurs when homologous chromosomes are coming together and they form their synapsis or synaptical complex. And what happens is that some of the DNA or genes from one of the chromosomes will come over and become part of the other chromosome. And so instead of just inheriting your original maternal or paternal chromosome, you actually will inherit a new chromosome, which we call a recombinant chromosome, that contains parts of both of the original. And they believe that this happens because the chromosomes are trying to stay together during prophase and metaphase 1 when they're lining up. And so they come really close together, and that's where the swapping happens. And the chiasma is just where this is occurring, where they're crossing over. On an average, there's about 1 to 3 crossover events per chromosome pair. So that's quite a lot. And it just depends on the size and the location of the centromere. So if you have larger chromosomes, you're probably going to have more crossing over events. And so as a result, if we had this crossover event right here, if we had 2 crossing over events, then we're going to have some, in our gametes, we can have some recombinant chromosomes, which means now we have some more genetic variation, because this is kind of a unique chromosome because it contains genes from both original chromosomes. So that's another way that we create genetic variation. The third main way is by random fertilization. So this is kind of taking these 2 principles and taking it one step further, because right now we're just talking about the formation of gametes, but really we want to form a zygote when we fertilize our gametes with each other and see what happens. And so when we do this, there creates even more genetic variation. So let's use humans as an example. So each male or female gamete if you grab a random egg or sperm, is one of 8.4 million different combinations. And that's just using independent assortment, because as humans we have 23 chromosomes, and so if you take 2 to the 23rd to see how many different combinations you have, it's 8.4 million, which is a ton. And so then, if you have 8.4 million different sperm and 8.4 million different eggs, the likelihood that one of those is going to fertilize the other, and the combinations that could form with all of those is about 7 trillion combinations. So that's a ton, but then keep in mind, that's not even factoring in crossing over. This is just with independent assortment. So as soon as you factor in crossing over, then our number jumps even more, and so we have this huge number, and that's why every single human is completely unique. So if you're still having trouble picturing this, if we take our 4 different combinations that we found up here, we have 4 different combinations of egg and 4 different combinations of sperm. This sperm has a 1 in 4 chance of fertilizing this egg, or this egg, or this egg, or this egg. And that's going to produce 4 different combinations of chromosomes. If these are our zygotes, these circles over here. And you can do the same thing with all of these. And so when you come out, you have all these different combinations of chromosomes in these zygotes. And some of them overlap, but overall it's going to create about 10 different combinations with just 4 different types, but we have 8.4 million different types, so that makes the number that much bigger. So those are the 3 main ways that we generally talk about when we're talking about genetic variation in sexual life cycles. However, we need to always keep in mind mutations. Mutations are the original source of variation. They create different alleles. None of these would work unless we had a little bit of difference in the first place. Because if all of our genes were exactly the same, crossing over wouldn't really do anything, random assortment wouldn't do anything, because they already would have been exactly the same. So we need to have mutations that create the original differences in these, and then these 3 things are just going to further mix up the pot. And also mutations are important because that's how asexual organisms have genetic variation. So there is genetic variation that's possible in organisms that reproduce asexually, and it's because of mutations. And then finally, just keep in mind that mutations are really important for evolution. Genetic variation is something really important for evolution. So in further videos and in further units, you're going to learn about natural selection as it applies to evolution. And you'll learn that without genetic variation, natural selection wouldn't be possible. So that's why we kind of care about this anyway. And also it's kind of interesting. If we didn't have genetic variation, then we would all look the same, and that would be no fun. So we like our uniqueness, and so we can credit independent assortment, crossing over, random fertilization, and mutations to this. That's all for today. I hope the video was helpful. I hope you understand these 3 and mutations a little bit better and how they relate to genetic variation. Thank you so much for watching, and have a fantastic day.